Okay, everyone, we'll begin our session now. I'm Scott Seiler. I'm the Chief Scientific Officer for the DILIsym Services Division of Simulations Plus. And it's my pleasure to introduce the president of the DILIsym division, and that's Brett Howell. Brett was originally trained as a chemical engineer and transitioned into the world of quantitative systems toxicology and pharmacology modeling, and has been leading the DILIsym efforts for several years now. And at this point, I'm going to hand it over to Brett to give us the webinar.
Thank you, Scott, and thank you to everyone for joining the webinar today. We'll be discussing the release of DILIsym version 8A. We're very excited about this version and hope to share some exciting new features with you. If you have questions along the way, please send those in via chat, and Scott will read those at the end. We should have 10 to 15 minutes to address those. First, let me acknowledge our fantastic team here at DILIsym Services. Without this team working on DILIsym over the last year, this release wouldn't have been possible. So, shout out to our team. And of course, for those of you who have worked with us on consulting projects or will in the future, you can look forward to working with a great team of smiling faces. So, a quick overview of the agenda today. We will start with a quick introduction to what the DILIsym platform is and is used for. Keep that brief, and then we'll look at the DILIsym version 8A highlights, a few slides to cover the new features and options, and then we'll move into a quick 10-minute or so demo, just looking at some of the new things in the live software, and then we'll have some questions. So at Simulations Plus, as a family, we are subscribing to the concept of embracing QSAR or machine learning, combining that with PBPK modeling or prediction of exposure in a mechanistic way, and finally quantitative systems modeling. And for example, taking something like ADMET Predictor, combining it with our platform GastroPlus, which has a PBPK, expansive PBPK capabilities, and then coupling that with a lot of what we do here at DILIsym with quantitative systems toxicology and pharmacology. And of course, today I'll focus on the quantitative systems toxicology aspect, predicting unwanted or undesirable effects of our compounds. So at DILIsym Services, we have three primary programs. Our DILIsym program, of course, the focus of today, also programs in kidney disease and NAFLD-NASH. We've recently launched a new program in idiopathic pulmonary fibrosis as well, or IPF. So that'll be coming. DILIsym in particular is focused on predicting drug-induced liver disease. We're now in our 8th version, and it includes many mechanistic representations of hepatic function, sort of normal homeostatic hepatic function, and then perturbations thereof with compounds. And we've now run greater than 60 compounds through the platform, sort of in the prediction scenarios, and worked with many companies. So if you're new to DILIsym, you may ask, well, how can DILIsym help my organization? Well, a few quick bullet points. It can help predict DILI liabilities beforehand and save you money, save your company money, save you from large, expensive failures. It can help choose the lead candidate most likely to succeed from a drug-induced liver injury standpoint. And it can also help you communicate with regulators on safety issues with information that they have requested from others numerous times, and a platform that at least the FDA, for example, licenses. So it's a tool that can be used in a number of different ways. Of course, above all, our ultimate goal is to keep the patients safer who are taking our drugs and who are volunteering for our trials. So it can do a number of things for your company. The software really came out of and is originated from the DILIsym initiative. This has been a consortium of pharmaceutical companies with DILIsym Services leading the way as the coordinating member over the last several years. It started at Hamner Institutes and is now a DILIsym Services consortium. And we have had 19 major companies participate over the years. The companies have not only brought sort of financial capital to the table to help spur development and new science, but also scientific capital through expert scientists and data that's been collaborated as well. So this is the format in which DILIsym has been developed and continues to be developed. And if you're interested in membership, just a few quick notes on benefits thereof. Members receive access to the software, of course, receive a PDF copy of the equations, receive training and materials, including some free one-on-one training or specialized training for your organization or situation, access to our online documentation site, which I will show today briefly, and discounts on consulting. But above that, there's also a chance to vote on what goes into the software next. So sort of a unique aspect, you get to formally vote on the items that have been put forth by other members and by our team as potential nice-to-haves for the future, upgrades, updates to potential areas in terms of the science. And finally, it's a great networking opportunity. A lot of good scientists from modelers, toxicologists, clinical pharmacologists, and others, physicians as well, have participated over the years and networked and learned a lot about DILI. If you're more interested in sort of a licensing option, we have that option now available. You can see sort of some of the capabilities of the platform on the right, kind of what it looks like. We not only provide commercial licenses, but we hope everyone can help spread the word that we offer academic licenses and regulatory licenses as well. We really want to spur sort of a research environment with DILIsym and also have others test and utilize the software and publish with it and help us make it better, improve it along the way. Finally, the DILIsym initiative includes an expert scientific advisory board, a really top-notch group of experts in different areas associated with drug-induced liver injury, and also a global group. And they've really helped us at a high level steer kind of the direction and make sure that we're addressing the key needs in this field of drug-induced liver injury as we go forward. So DILIsym itself is actually a quantitative systems toxicology software that includes 4 species: mouse, rat, dog, and human. It includes a number of different submodels or key components or mechanistic areas that you see kind of summarized in the diagram in front of you. Those include sort of drug exposure, which we handle a lot of times with GastroPlus, and we bring that information into the software. And I'll talk more about that link in the future here in a minute. But also we have a capability to do that in DILIsym. We focus on mechanisms such as oxidative stress, from reactive metabolites or stable metabolites, mitochondrial dysfunction directly, perturbation of bile acid homeostasis, disruption of the bile acid phospholipid ratio, and the disruption of the cholangiocyte life cycle as well, in addition to the hepatocyte life cycle. And of course, biomarkers are a big component of what we do, and inflammation. So of course, each of these represents a large group of equations and parameter values behind the scenes that are used in the software to make predictions. We've represented over 60 compounds in sort of the prediction type scenarios, and we've had greater than 80% success with predicting the outcomes for those. But of course, we want to continue to improve the software. So these are some of the things that the current software development driven by the consortium's goals are focused on, including immunology and the adaptive immune system, an improved interface and environment. Again, I'll talk more about that in a moment. New populations or simpops, including not only disease groups, but larger groups, and connecting those with real-world patient data as well, and also cholestasis, which I'll talk about today. So there are a number of new things in addition to this list. Sort of the last bullet point there is we are working closely to improve the in vitro data that we feed into the software too. So we have a great system today, but we believe that some of the new state-of-the-art systems may provide more predictive inputs. So we're working in that area. So how does the software work? Its most sort of basic description is, of course, we're using the software to predict risks, to understand mechanisms that might be involved in that risk. One component you need to run the software are specific in vitro data. Excuse me. Sorry about that. Had a cold here recently. You need some specific in vitro data to feed the software. That includes high-content imaging, assessments from the Seahorse XF analyzer, and also some transporter information. You need predictions of exposure, in our case, in particular for the liver, And finally, you need to know something about your goal, the clinical study or the preclinical study you're trying to simulate. So, of course, the species, the dosing protocols, the fasted or fed state, and potentially the patient population as well. And then all of that gets fed into the software, and out comes the predictions. We've used the software now in the context of a number of projects that have been involved in communications with regulators, largely the FDA, but also the EMDA and PMDA. And, of course, as I mentioned, the FDA has purchased licenses as well. So thus far, we've seen that regulators are very interested in this type of software and what it can bring to the table in terms of sort of the weight of evidence for or against the safety of a given compound. So I wouldn't say regulators make their decisions solely based on this type of mathematical modeling, but certainly they've been very interested in understanding what it suggests and, of course, what the data inputs say and how that fits into a larger data package. And in the future, I think everyone can look forward to a new platform for both DILIsym and GastroPlus. We're working on refactoring both of these products, and we will offer DILIsym as a standalone in a refactored, almost C++ type environment. We plan for it to be more modern and faster. Same with GastroPlus, but we will also offer DILIsym as a module of GastroPlus. So that will be coming down the road. It's another effort that's underway. So now let's move into looking at some of the DILIsym version 8A updates that have gone in. We've got 10 new exemplar compounds. They have sort of varying clinical presentations, and they do include some compounds from collaboration with GSK. We have a new cholestatic liver injury mechanism I'll talk about, a new oxidative stress adaptation framework, very important for this idea of adapting to injury. New populations with variability in bilirubin or new simpops, a new biomarker, GLDH, and also some new immune system components and bile acid components that I'll talk about more. I do want to highlight the simpops updates here because they're quite important. As a result of some of these mechanistic updates, we had to do quite an overhaul of our simulated populations in this version 8A. So many of you who've used DILIsym in the past, you'll notice when you open the software that the simpops have changed quite a bit. And many of the older ones have been removed because they weren't compatible with the older version of the software. So just look for that. I'll mention it briefly on the demo as well. We've added some new sort of graphical interface features, and I'll go through those in the demo. And we've also updated the MATLAB version that we've compiled this on. It's now in 2018B, so the latest, greatest version. So to expound a bit on these, first of all, these 10 new compounds include 7 that came from a GSK collaboration where we and GSK simulated these compounds or represented them in parallel to test the software and test data, the nature of the data inputs and sort of how different types of data packages would lead to different predictions. And those compounds are included. Some of them are still blinded. So there's a compound A, B, etc., but then some of them are not, as you can see at the top. We've also got 2 new tyrosine kinase inhibitors and valproate included. And there's really at least a couple of benefits to these types of additional exemplar compounds. Of course, you can use them for training and learning as you're going through the software. You can also, from time to time, use them for DILI-related DDI predictions. So the more exemplar compounds we have in the software, of course, the more DILI mechanism, DDI type scenarios you can predict for new compounds that are being developed and being combined with compounds that are on the market. Next, we've looked at and added to the software a cholestatic component. So MDR3 inhibition, which is a transporter in the liver, can actually lead to a disruption of the bile acid to phospholipid ratio. And so we wanted to represent this in the software and at least as a framework so that we can start prediction in this area and understanding and exploring the science more. And so we represented phospholipid homeostasis. That was a new component. Represented the drug effects on MDR3, added the cholangiocyte life cycle. That cell type was not there previously. And we also represented the bile acid effect and the influence of sort of MDR3 on the ratio of phospholipids to bile acids and how that could lead to cell death. So you can see here, for example, if a drug goes into the cell, you get inhibition of something like BSEP, but now, in addition, MDR3, you can disrupt this bile acid to phospholipid ratio and get that cholestasis component, in addition to kind of a typical hepatocellular component coming from bile acid buildup. So the cholestasis submodel was constructed as shown here. We have different stages of the life cycle for the cholangiocytes. We've also represented the ratio of bile acids to phospholipids, and this kind of added that as a variable in. And finally, of course, we have the downstream biomarkers associated with this process. So we've got GGT and serum alkaline phosphatase, and we've really based initially this relationship between cell death and the bile acid to phospholipid ratio on some human liver transplant patient data where the bile acid to phospholipid ratio has been measured and cholestatic biomarkers were measured. So it's an initial start at getting this relationship calibrated, and we'll be working on validating the model over time. And this is just a theoretical example here on slide 20 to show that on the right, as you inhibit MDR3 in the software, you can get a loss of your healthy cells and an increase in apoptotic cells, and as a result below, you see alkaline phosphatase and GGT increase. So these are kind of some of the sensitivity analysis we've started with. We've also started looking at some specific compounds, like, for example, itraconazole, and how they would affect and lead to cholestasis. So that module is still sort of in development and validation, but the framework is there and available for use. So in addition, I want to talk about NRF2 and its response on oxidative stress. We incorporate an adaptive response and have in the past from NRF2 on glutathione synthesis and homeostasis. However, we also know that it drives up glutathione and other enzymes for the purpose of clearing oxidative stress, not just for the purpose of clearing reactive metabolites. And so, and of course, there are other enzymes like pyridoxine and superoxide dismutase and others that are being upregulated through this type of pathway, NRF2. And so what we've done in 8A is add a framework that allows you to explore this feedback and adaptation mechanism. And here's some data, for instance, in rats and mice showing that we have, in preclinical data, there's in vitro data as well, but especially preclinical data showing upregulation of some of these clearance pathways and how that could affect and lead to adaptation. And we followed a similar sort of strategy with our mitochondrial adaptation framework. We put a framework in initially without the parameters. We worked over time to validate that and then added more validated parameters. And even today, that sort of mitochondrial adaptation framework is still being refined, but there are parameters in there that have been validated to some degree with some clinical data and preclinical data. So we'll continue to look for more data to validate this submodel or this new feature. So this diagram here is a quick schematic of our oxidative stress, reactive oxygen and nitrogen species diagram, and it shows that currently it's simulated as a balance of the species. So it would start at 1, for instance, for a homeostatic healthy person, and you have clearance of the oxidative stress and production of that oxidative stress. And notice now that the clearance in sort of the prior versions was just sort of a single clearance term. It's a saturable clearance term, but it's not dynamic in the sense that NRF2 pathway is affecting it. In DILIsym version 8A, this framework still applies, and when you open the software, by default, it's going to kind of continue to use this framework. However, there's a way that you can now turn on a new enzymatic clearance option. So there would be an enzymatic clearance versus a non-enzymatic clearance of oxidative stress. And now that NRF2 signal that we have already included and we previously used only for glutathione homeostasis is now being used for the enzymatic clearance. So to enable this new feature, you go into the species parameters, into the NRF response adaptation subgroup, and there you can turn on the effect, and not only that, but you can also parameterize quantitatively the parameters for that section. So you can explore, perhaps you can fit those parameters to a situation you've seen within your organization where oxidative stress showed up on the in vitro panel, and there was adaptation in phase 1 studies where patients, ALT came up and then it went down over time. And you can calibrate to that and say, well, this perhaps could be the mechanism. What would happen if I increase the dose, et cetera. There's also an output panel specific to this ROS adaptation mechanism. So that allows you to look at many of the key outputs that have been added to the software, for this approach. And just a quick example of how this can be used. In this case, it's a very subtle example, but it's just purely fictitious to show the point here. So you notice on the left how we have an increase in oxidative stress at let's say around 100 or 80 hours, and this is a single dose of a compound, and we have a nerve adaptation signal below. So now we have the system responding and nerve kicking in and trying to upregulate a number of things. But on the right, what we see is a little bit different scenario where actually the drug was given beforehand and then at a lower dose, and then there's the same dose. And what you can see is that because of that loading dose adaptation earlier, we now have a reduced oxidative stress. So if you look at the red dashed line, you'll see the oxidative stress peak has gone down from above 2.5 to below 2.5. And we see a similar type view with ALT here at the top. So it goes from above, the fold change from above 15 to below 15. And really, depending on the timing and the size of the dose beforehand and the number of doses, this effect can vary in terms of how much or how little it affects the adaptation. But nonetheless, it's very much in line, at least practically speaking, with many of the adaptation patterns that we see clinically, including with acetaminophen, where people have been able to become tolerators of really high doses. In addition, we have a new bilirubin simpops that allows for variability in response to perturbing the bilirubin processing pathways. So by this, we mean the pathways responsible for the transport and the metabolism and the processing of bilirubin, as opposed to bilirubin as a biomarker of liver dysfunction. So many times, bilirubin can be elevated, and not because of injury, but because of disruption of the processing. And that can become a rogue signal. It can really confuse the discussion from a safety standpoint, especially when you have concomitant ALT elevations that are minor. And so we see this quite a bit. So this new simpops is focused on giving us variability in some of these pathways. What you can see on the right is the distribution of the sort of serum total bilirubin at baseline of our simulation in red and the data set in blue. So it's a good match there. And then we've also included subgroups of different UGT1A1 phenotypes. So we've got normal, intermediate, and slow for those three for UGT1A1. And they have different sort of ranges, as you can see here, of UGT1A1 function that leads to different total serum bilirubin at baseline. And you can see the data compared to the simulation. The data is in black and the simulations are in gray here. And you can see that with the slow metabolizers, we have an increase in serum bilirubin very much consistent with the data. So this allows you to look at special groups and how your drug may be affecting special groups as well. Now, just to illustrate this, we have an example here on efavirenz, and this is a drug, a monotherapy that significantly reduced bilirubin, actually. And if you look at the gray bars compared to the black bars, you'll see that both for total, unconjugated, and conjugated bilirubin. And so the drug reduced all three of those types of bilirubin, the total, both conjugated and unconjugated. And the question is, what are the exact mechanisms involved in that? And so what we did was we ran a simulation where we included induction of, in addition to UGT1A1, which is known to be important there, we included induction of MRP2 and also OATP1B1. And we looked at some different factors. And of course, this is a perfect example of how a model can be used to tease out different proposed and hypothesized mechanisms. So with UGT induction alone, we saw that the total and unconjugated bilirubin looked good in the simulations compared to the data, as you see at the top with the two green checks, but below, with the conjugated bilirubin, we didn't see the decrease. When we looked at UGT and MRP2 induction, both of those together, we saw simulated outcomes that look consistent with the data on the far left. And then finally, when we looked at OATP induction, we saw that as well. So it just leads us to, okay, here are some mechanisms that seem to be consistent with this data and others that don't, and now we can follow up on that with further study. Of course, we can also use this whole new, our bilirubin processing model and also the simpops for scenarios where we want to predict a priori that a drug will increase the bilirubin and be aware of that going into the safety discussions. Next, we've included GLDH in version 8A. GLDH is a mitochondrial enzyme. It is primarily found in the liver. And the nice thing about GLDH is that it can be used in situations where ALT is confounded. So, for example, things like muscular dystrophy, where ALT may be coming out into the serum due to other conditions besides liver injury. And so GLDH has been a favorite of many of the consortia recently in terms of moving it to closer to another gold standard liver injury marker. And so we took advantage of this and put it in DILIsym so that others can use it now as part of their outputs and connect to their clinical results. The structure of the model is very much like acetaminophen, ALT. It includes cell leakage from the cell source as the cells are necrosed, or in some cases apoptosed, depending on the level of apoptosis. It includes release into circulation and clearance. And so it's sort of like a PK model for GLDH in a way. And we know GLDH correlates well with ALT, so we took advantage of that for some of the parameters that we needed. And here on slide 31, I have an example of how we optimize the model and the results. So we, first of all, many of the parameters, such as the baseline levels, the half-lives, these things were able to come directly from literature. And then finally, we used the GLDH to ALT concordance in certain studies to optimize the remaining parameters, such as the delay times that we need to release from necrotic cells and otherwise. So you can see a good fit there by design between a situation where ALT was increased and so was GLDH. And we did the same for rat. Here you can see some of the rat data where we're able to line up nicely with the GLDH levels and the ALT levels concordantly and optimize those parameters. So we have a model now that's reasonably well optimized for rat, human, and mouse. Dog, we have a model there too, but it's not quite as well optimized. And now we will validate this model over time, looking at more and more human data. And of course, we encourage you all to do that as well. We've included neutrophils now as well. So here you can see after an acetaminophen overdose in mice, you can see, or a large dose in mice, you can see neutrophils accumulating, and our simulations now include this new cell type. In addition to the accumulation of neutrophils, we have the mediator production profiles by the neutrophils. And so, and with variability, in the data, which is always the case with the data in the immunology space. So, what this really allows us to do is focus on some of our future applications where neutrophils will be important, such as some of the things we're adding on the immune system side in the adaptive immune area, and in updates we'll be making to our innate model as well as we go along. We've added a new bile acid renal clearance and ASBT mediated gut reuptake feature. And this really allows us, first of all, to address a disconcordance we had in the past between plasma bile acid levels in the simulation and those that were measured. So we added these. We knew these were important. We added these in. The key here is that because we added these features in, the entire bile acid homeostasis model had to be reoptimized. And this is why it's very important to understand the simpops have changed. And the simpops are now, slightly different. We're testing them against a lot of compounds. We believe that they're valid and good simpops, and so was the old one. But they did need to be replaced because it had to be reoptimized for this purpose. So this was one of the key drivers for that process. And here on slide 35, what you can see is that above in the plot on the top right, we have the plasma bile acids from the simulations with the black dots, and then we also have in red, the data in black dots, and then in red, the simulations. And notice that the plasma bile acids on the far right are, from those who've been treated, are extremely high compared to those who were treated. We see a slight elevation in the paper. We see a large elevation in the simulations for version 7A and the older versions. But down below for version 8A, now we see a much better concordance between the increase in plasma bile acids from this particular publication with this particular drug, which I believe was Bozin. So, yes, it was Bozin. So, given over 30 days. So, this is improved the human plasma bile acid model. The rat plasma bile acid model, when we actually updated to add some of these new features, it slightly became less accurate with the plasma level. So we're still working on that and refining why for the rat some of the parameters might need to be different. But at least we know that the framework makes sense and that it needed to be updated. And we prioritized, of course, the human data. So now at this point, I'd like to move into a quick demonstration of some of the new 8A features. So I'll start here by creating an example sim single, and I'm just going to make a blank one. Call it example. And if you're interested in some of the new compounds that have been added and sort of investigating those, what are the mechanisms, you can find those under the drug tab. And for example, if I scroll down, say close to the bottom here, I can find the new valproate compound. I can click the mechanism tab, and there you'll be able to find many of the key pieces of information on what's driving the valproate mechanism. And a lot of our work for valproate in particular has focused on the mitochondrial effects that you see from the in vitro systems, and we do see adaptation to valproate treatment clinically, and we've been able to reproduce that in our simulation. So it's a very interesting molecule from that sort of adaptation perspective, and some of the key parameters and things that are needed from that side, and also concepts that apply to other compounds where you see that. For some of the new simpops, just to open the simpops here, I'll show that when you open the simpops, you will indeed see the list has changed quite a bit. So many of our 7A simpops and the older ones are gone, and now you'll see, for instance, the human v8A1 simpops. So this has been updated, has some of the parameters included are new values, different values. One of the things that is important to know here is that you may want to use the simpops feature, which of course has a batch feature, so you can run multiple simpops together and run a full population. But in addition to that, you can also run subgroups of those populations, and those we call sim cohorts. And one of our new features that has been added as of DILIsym version 8A is that you can now do a few more things with our sim cohorts generator. So before, you could add indices, for a given simpop. So let's say, for example, I want my source to be the UGT1A1, slow metabolizers. That would be the 8A10 simpops. I can add individual indices, and I can pull out certain indices and you could do that certainly in the past. It's important to note here that if you were picking certain individuals out, you could note those individuals, and you could also go over into GastroPlus, which has a DILIsym mode capability, and you can put those same indices there and pull out the same individuals for your PK traces that you want to pull into the simulation. So there's you can use the index list in both pieces of software. But now we also have the ability to shuffle. So if you want to change the order, and the reason this is useful is if you're using, let's say, for instance, GastroPlus outputs, which include individuals 1 through 100, and the PK for individuals 1 through 100. If you import that PK, it's going to line up with individuals 1 through 100 in the software here. And so, if you wanted to, for instance, change the order of the concordance between the PK and the individuals here, you could do that by shuffling that group. And sort of randomizing it. And again, if you want the exact individuals from GastroPlus with the matched body weights, you would pull that index out and take it to GastroPlus. So I'm actually going to, and the other thing that's really nice that you can do now is you can create subgroups. So instead of just being able to shuffle 100 individuals, for instance, I can create a subgroup of 10 individuals. So I'm going to create a sim cohorts here for this particular simpops, and it's going to put the new group in the simpops folder. And so once you create the simpops, one of the key questions is, well, how do I know where each individual went, like how the shuffle worked? And also I might need that information to use in another application to try and pull out similar individuals or to track down why simulation results have changed. And so you can navigate to the simpops folder. So I'll do that here. You can use a little trick where we pull the local path, and we can back up and go into the simpops directory. And there we should see a new subgroup file, an Excel file. In addition to the new mat file that's been created for this new subgroup, we have an index list here. So this is the index list for the individuals that were pulled from the old simpops, the default simpops that I pulled from. And here's the new index, of course. So now, instead of being number 82, has been shuffled to be number 1 in this group of 10, and so on and so forth. So now you have that information. You can use that information as needed. Another feature that would be nice to show is our new the ability to work with sim singles in a more user-friendly way, let's say. And for those of you who have created a number of different sim singles and tried to work with those sim singles in sort of a batch type way, for example, maybe you create a parameter value, a sim single with a certain parameter, and then you realize later you've got 30 sim singles and you need to change it across all of them. It's quite difficult in the current setup unless you're using MATLAB and you go in and use behind the scenes. But what you can do now is, let's say, for instance, we have a new sim single simulation name, and I save that. You'll note that I now have 2 sim singles saved. Both of them are just blank examples. But the new capability we have is the ability to export sim singles to Excel, alter those sim singles, and then bring them back into as new sim singles. So you can export an entire, let's say you have 30 sim singles, you could run this export feature, and all 30 will show up in the Excel file. You can make your changes. Maybe you want to change one parameter across all 30. You can even go in and add new columns and add new Excel files. Add new sim singles. And then when you go back to the file menu and do import, it will pull all of that new information in and recreate those sim singles. So this is a much more user-friendly way to manage these sim singles. So once this converter completes here, I will open the file and show you the way that the parameters are arranged. Actually, the sim singles are in different columns, and then the parameter values, as you see here in J and K. And if we expand this a bit, first there's some information at the top that really shouldn't adjust or change. That is from the sim single as it was created. You can adjust it or change it, obviously, when you're making new ones. And then below, we'll see all of the parameters from the software. So it starts with the species data, and actually will go down all the way to through the drug information or the compound data, all types of inputs. So you can change anything you want in terms of the values here. These are the main columns that you should be focused on, the values for those parameters. And you can also add new sim singles, and then you can save this. And once it's saved, go back to File and import the sim singles, and you can do that in a batch type fashion. The last feature that I'll show here is related to our sweep feature. So we've added a nice feature for what we call specified data. So specified data is when you want to pull simulation results from another application. Maybe you have a different PBPK model that's predicting your liver concentrations from another software and your circulating concentrations, such as GastroPlus or others. Or maybe you're using actual preclinical data to drive your concentration that you're using for your predictions. Either way, you may be putting those things in and importing them into our specified data feature, and we have, of course, a whole training section on that. But one of the things that was difficult in the past was if you imported that specified data, let's say liver concentrations for 100 individuals, it was quite difficult to alter that from within DILIsym. So you would have to go back to Excel, make changes, and sometimes you would just want to do quick and dirty analyses. And so what we've added now is a specified data sweep feature. So here, basically the sweep numbers, the lower and upper limits, for example, 0.1 and 10, are scaling factors on the specified states that you put in. So here you see it's blank because I haven't put any specified states into this sim single. But if I had put, let's say, liver compound Y in as a specified state, it would show up in the dropdown. And then by selecting these numbers, I'm telling it I want to scale every profile by 0.1 all the way up to 10. And I could actually do a more granular curve. I could say let's do a scale of let's scale 10, and then as I hit this plus sign, it would show me a table, of all the different scaling factors I'm going to use. So, for instance, if you have a median, specified data spreadsheet, and then you want to scale it and go twofold or 50% reduced exposure, you can do that. And you can do that, of course, with any state, but it's really most applicable for the pharmacokinetic inputs that we often use specified data for. So that's a nice new convenient feature. Lastly, I just want to quickly mention our documentation website. So if you log into the DILIsym.help.com, you will see our DILIsym documentation website has been updated for this release. So, for example, here's our new summary diagram with cholangiocytes added in, and there are a number of things to be aware of within this new updated version. So, the user guide, for instance, has been updated to include information specific to DILIsym version 8A, like some of the new features I just mentioned, but also others. So you can glance through that and find the new areas you might not be familiar with. In addition, our notes section has been updated for some of the new mechanisms and some of the new information. And we also have a feature included. So let's use hepatocyte life cycle as an example. Each submodel or each area will have a diagram showing a depiction of that model. We have design rationale and descriptions of the literature used. And one feature that we've updated for version 8A in particular is adding a different view for the differential equations. So now you can see that, for example, with the hepatocyte life cycle, there's a viewer here. You can open those, and in LaTeX format, you can look at the differential equations and sort of map those out if you're interested, or at least just try to understand, well, how is this particular component being represented? And in addition to those equations, of course, we also have documentation related to our simpops and our sim cohorts. So there's overviews here for both our full populations and our subgroups, but also beneath each there's a PDF file, and you can download that file, and it has all the details in terms of what parameters were varied for that population, what is the purpose of that population, and what are the literature sources that we use to underpin the parameter variation. Last but not least, it's important to know that we have a whole section of training videos on this documentation site. So essentially, a whole DILIsym course is laid out here piece by piece in YouTube-like videos. We have introductory videos that range from introducing DILI as a clinical from a clinical side to the software environment. We have data collection, the setup of the software, and also many of the features highlighted here. And then as well, a couple sections on how to use GastroPlus to drive DILIsym and how to do PK modeling in DILIsym if you face that challenge. So we also have some of the user review sessions that we do as well. So I think I have a couple more slides to cover here, so let's switch back. We're almost wrapping up, and we should have time for questions. So some key dates we have coming up in 2019 for DILIsym. One is we have a 3-day training course coming up in February, and it's in Redwood City, California. It's a 3-day course. We're offering sort of 1-day attendance or 3-day attendance. The first day will focus on sort of a general introduction. It'll be mainly slides or demo. The second day will be step-by-step compound representation, and it'll be more hands-on. And then the third day will be some of the advanced features and looking at some details of those features like simpops, our monitoring feature, which allows you to implement clinical monitoring into your simulations to try and determine how that would impact your safety outcomes, our optimization module, which relies on the genetic algorithm, and our specified data module as well. So we're offering, importantly, we are offering remote attendance and on-site attendance at the Seaport Conference Center. So feel free to sign up. For those of you who might be doing the advanced GastroPlus courses that week at the same center, but not anything earlier in the week, you could come to the course there. So feel free to contact us if you're interested in attending the training course. We also have a DILIsym Initiative Consortium meeting coming up in late March. If you're a member of if your company is a member of the initiative, feel free to attend that and see some of the updates. So with that, I want to thank you all for attendance. Feel free to contact us at our websites and reach out to me directly. And I'll stop and let Scott read a few questions. Scott.
Thank you for the presentation, Brett. We do have a handful of questions here. in a couple of categories. Let's see, I'll start off. The first question is, I'm trying to make sure I've got it right. Okay, so I would describe it as a general question related to the licensing of DILIsym. So would you mind just reviewing some of the licensing options again, with the focus from the point of view of industry? Does industry need a license for DILIsym? Can the software be tested?
Sure. Yes, we do require licenses for users to use the software. However, we do offer trial versions, so just contact us and request a trial version. We can make those available. We have two different formats for making the software available. One is through the DILIsym initiative, which I spoke about. So if your company joins the initiative as a member, you get two global floating licenses to the software. And those are annual licenses, so as you renew your membership, your licenses renew with it. The other option is you can just purchase a license. You can purchase single licenses or larger packages. As you add licenses, you get discounts on the subsequent licenses. So both of those options are available. There's some differences in sort of the benefits you get in one case versus the other, and some of the opportunities as a member versus a non-member, but both of those make the software available to you. And of course, we have a number of training options for both members and licensees. So.
Okay, moving on to the next question. This is sort of a high-level question, kind of really leveraging our group's experience using DILIsym. So what is the major mechanism of DILI that we have observed in our simulations with the over 60 compounds that we have evaluated thus far?
We have really seen the mechanisms responsible for the ones that are sort of, let's call them the positive controls, the ones that lead to DILI, span the gamut. And I think I can make a few comments on interesting aspects of the mechanisms, but I can't say one particular mechanism is the primary mechanism. First of all, I think what we've noticed is that it's multifactorial. That's been a big learning. When we started, we thought of this in some ways as, some compounds do this and others do this. And we realized right away that it's actually much more multifactorial. That is, of course, the power of having a quantitative systems platform. And when you, and in that vein, we've seen a whole number of scenarios. So, for instance, we've seen compounds that do cause injury from one mechanism, and the other mechanisms tend to be relatively less impactful, and maybe even doesn't do anything in the in vitro systems for those mechanisms. In other cases, we've seen that compounds can hit the mitochondria and also lead to bile acid dysfunction, and that interplay, the multifactorial nature of those two things happening in tandem. Sometimes one is a stronger influence than the other, but then both of them together push certain individuals over the edge where they have some DILI response. So bile acid toxicity and mitochondrial dysfunction together, those two have come up a lot as an interaction effect. And then, if we've run more compounds, we've also recently seen a number of scenarios where all three mechanisms are involved. So without any one of the three, you have a lack of response. And actually looking at the responses in isolation for the three mechanisms, you would think that the compound was clean. But when you put it all together, especially in cases where there are large doses of compound or long-term dosing, you see some toxicity, some signals. And so the last comment I would make on that is that we've also seen some interesting interplay between the different molecular entities for a given drug. So the parent, in other words, versus the metabolite. So in some of our cases, we simulate parent and metabolites and their effects, and you can certainly do that in our system. And we've seen that the metabolite does A and the parent does B, and together they cause the problem. So it really gets quite multifactorial, relatively quickly. And that's not even to speak of the fact that we've started to look more at DILI drug-drug interactions. And so one compound and another compound, and both have metabolites, and all of those entities can be doing different things. It's interesting to look at that type of problem as well.
Okay, the next question relates to the types of species that are represented within DILIsym. You mentioned that the human, the rat, the mouse, and the dog are all represented. Do you foresee additions of additional species in the future, such as cyno, pig, or others?
Yeah, that's certainly a possibility, and in large part, the selection of the species will be driven by the DILIsym initiative and the votes that the members cast. And thus far, I would say they have deprioritized the additional species over additional science and other key updates, such as focusing on the in vitro panel, immunology, and some other things. So I think what's been interesting over the years is that we've seen really more of a focus on using in vitro data from human cells, human cell types or cell lines or primary cells to predict human outcomes. And that's really where we would hope that the software is used, and there are some definite applications of the preclinical models with the human model, but I would say that has not been as big of an application area for us, and so that's probably why more species haven't been highly prioritized at this point.
Okay, two questions in the category of bile acids, and I think we can kind of aggregate them. One is that, let's see, does DILIsym focus on specific bile acid species? That's part A. And part B is, does it also include any of the metabolites of bile acids, such as sulfation and other conjugates?
So DILIsym includes the current bile acid submodel includes chenodeoxycholic acid, or CDCA, and lithocholic acid, LCA, and it includes the conjugates of those specifically, sulfation and amidation. It also, and a number of metabolites. It also includes kind of a bulk bile acid pool for a lot of the others. And so the origin of that is that we did in vitro studies to determine what were the most toxic bile acids. And those and their products were ones that kind of popped out as having, pretty overt effects. So we specifically represented those, and those are the ones that have the mitochondrial uncoupling effects that lead to the toxicity in the software. And then the bulk bile acid pool, is there as well, but it's having less of an effect on the toxicity and more of an effect on the homeostasis of the bile acids themselves. So those currently are the ones that are represented.
Okay, two questions about the software. One is to please summarize the import PK data feature. The other is a question about whether, since this software was originating in MATLAB, is it possible to use MATLAB to make adjustments to the code?
Okay, so the PK import feature is simply a feature where within the software you select the specified data option. That's what we typically call it, specified data, and you can build by selecting the state variables from a drop-down menu. You can build a template that you can paste in data or simulated results, information that then overwrites the state variable equations in the software. So the typical example is you want to import simulated or predicted liver concentrations and plasma concentrations, or a number of those. You can build a template with that information in it and feed that into the software and say, I want to use specified data, and that overwrites those equations. And so basically you're starting from that information and driving everything else. For example, if you're doing it with exposure, you're driving the responses from that exposure. Now, the benefit of that is you have the freedom to use a lot of different information in driving simulations, and it also helps for exploration and model building at times because you can do it with other state variables besides just pharmacokinetics. In addition, we've Gastro Plus 9.6 and now 9.7 to come out in the coming months will include, for instance, the DILIsym version 8A simpops and the version 7A simpops. So you can use Gastro Plus, and it will actually create that template for you. You can select the right simpops that you're going to use in DILIsym. You can create your simulated population in Gastro Plus and export that PK information. It spits the Excel file out automatically, and then you just load that right into DILIsym and use it. So that for Gastro Plus in particular, we have an even more convenient connection. The caveat with that type of connection is, you break the loop between the PK and the PD, so you don't have the feedback. But in many cases, it's less important if you're screening for an effect, if you're looking for a response as opposed to a real serious or significant injury. And the second, so that feature is available, and again, we have videos and training offered to kind of go into more detail. It's quite, you can get quite complex with how you use it. The second question, can you modify the code? So the software is delivered in a compiled application, so modifying the code is not available, although there are a number of different parameterization options, as I kind of showed with the NERF2 adaptation to ROS, where allow you to kind of reconfigure it the way you want to run the simulation. In addition, as a member of the consortium, you get access to, as I said, a PDF document of the equations, whereas for those of you who license it or your members, you can also get the equations as I showed on the website, sort of in piecemeal. And finally, if you want updates and things, you can propose those, as part of membership, and we can make those to the equations.
Okay, I think we have time for 2 questions. The first, the second to last is, what is GLDH, the biomarker GLDH?
Glutamate dehydrogenase. It is again a mitochondrial enzyme, highly focused, concentrated, I should say, in the liver, and so it tends to be very liver specific, and even more so than ALT, which is considered to be a relatively liver specific enzyme as well. But you do see some of that in muscle. So.
Okay, the final question, then we'll wrap up, is basically the general question is, do individuals who are not members of the DILIsym initiative have access to the documentation and training videos?
Yes, as a licensee, you receive access to the documentation site, all of the videos, the information there, and you also the www.dilisymhelp.com is also where you can download the software and install it. So both of those are open to the licensees as well.
And is this information available to individuals who have trial memberships or do not have any licenses whatsoever?
Trial licensees can view the information on the website until the trial expires, and academic licensees and government licensees can also view it during their license period. So it's available to all of the above as long as the license is valid.
Okay, I apologize. There were a few other questions that I'm afraid we did not have time to answer. But as you know, we frequently present at conferences, and so we make ourselves available, and hopefully we'll be able to answer your questions in person in such instances. Of course, you can contact us in a variety of ways as well if your question wasn't answered. So with that, I'd like to wrap things up and say thank you very much to Brett for, like I said, a fantastic presentation. We appreciate your participation and interest in DILIsym, and we look forward to future webinar participation as well. So thank you very much.
Yep, thanks for joining.